Good evening. Welcome to the first Voices of Discovery speaker for the 2024-2025 school year. At this time, I would like to ask everyone to please silence all their mobile devices, and I'd like to ask everyone to please stay seated until the end of the talk. I would also invite everyone to stay seated through the brief question and answer session that will follow the talk. During the question and answer session, we invite anyone to come down and ask a question through these mics. So, my name is Tiberius Brooks. I'm an environmental engineering major here at Elon University, and over the past year, I've had the privilege of working with Dr. Bethany Brinkman and our Elon Facilities Management to help design and get ready to replan the water systems in the historic neighborhoods. So, Dr. Sanders, he earned his bachelor's degree in material science from University of Wisconsin, Eau Claire, and he went on to earn his PhD as a Rhodes Scholar at the University of Oxford. Dr. Sanders is the former director and now consultant with the Venture Studio at Southern Research, where he worked to develop companies that provide therapies related to oncology, neurodegenerative diseases, and more. Dr. Sanders is now the co-founder of Arrowhead Labs, where he works with university research institutes and precision health leaders to develop innovation strategies and produce and implement new precision medicine tools. Today, Dr. Sanders will share with us the journeys of two technologies that are being developed by companies he consults with. The narrative will highlight scientific challenges associated with these technologies and their potential in reducing costs of medical treatment. So without further ado, please help me welcome Dr. Tayo Sanders.
Thank you so much, appreciate it. All right, fantastic. Well, hello, everyone. Thank you so much for being here tonight. I really just have to start by saying thank you to Elon for hosting me first and foremost, to Jen Hamill for inviting me here. Wouldn't be here without her extending that very gracious invitation. And thank you for all of the students and the faculty that I engage with today, your time, your attention, your questions. I was sharing this before, but not only do you have a beautiful campus, but it reminds me a lot of UW-Eau Claire, my alma mater. It's a campus focused on undergraduate research and really close collaborations between faculty and students. And that was just in the air today. I could feel it in the environment. So really special place, and I'm so happy to be here. All right, let's talk a little bit about reimagining drug discovery and manufacturing. If I can get the clicker to... All right, I might use the laptop for now. Let's see if this will go. Oh, there we go, now it's working. All right, fantastic. So I'll start off the talk just speaking a little bit about myself, giving my introduction, my background, what's my story, right? I'd love to then walk through a little bit and generate a kind of a shared framework to ground us in understanding a little bit about what's going on with drug prices and drug development today. Why does it seem to be getting more expensive? I'd love then to walk through two technologies that I've worked with, invested in, and consult with that are really changing this paradigm. I'll walk through some of the scientific principles. Forgive me if it gets a little technical, but we'll also make sure to not skip over some of the cool data that we've generated, and in particular the impact that these technologies could have on the real world and on human health. And then we'll leave some time for just to share a few reflections, and of course, happy to answer any questions. So let's begin. My gateway into science. I'm from Wisconsin. I lived in Northern Illinois and Wisconsin growing up. My earliest memories, other than chasing around my sister with a plastic baseball bat, which she likes to remind me there's photo evidence of, are of asking just a lot of questions. Questions like, why was the sky blue? Why was the grass green? Was the moon actually made of cheese? I like to think that I got a little better at asking questions, but I was very lucky to have parents that supported this curiosity. When I saw Twister as a kid, I was slightly terrified, but I wanted to become a meteorologist or a tornado scientist, I called it when I was a kid. And again, I felt like that pathway was always encouraged. Anytime I wanted to be something different or a new scientific discipline that caught my eye, my parents were always there to support me. I pretty much begged them every year to go to the Museum of Science and Industry. I don't know if anyone's been there in Chicago. It's an incredible place. And then when I was 6 or 7, I got my first microscope, and that's where things got serious. I would say that every plant and bug in my neighborhood learned to fear me and that microscope. And as I continued my kind of scientific journey in high school, I really kind of struggled to pick something that I wanted to focus on. I was really interested in all of the scientific disciplines I'd come across. I was leaning towards biology, and I got to Eau Claire. And I'll be honest, I was more than a little naive when I arrived at college. I was a first-generation college student. I thought the only way to become a doctor was to go to medical school. I knew I wanted to be involved in science. I had no idea what that actually looked like from a career pathway. And I had no idea that scholarships like the Rhodes Scholarship or graduate school fellowship programs even existed at all or were opportunities that I could take advantage of. I also didn't really know that undergraduate research was a thing and that you could get paid to do it sometimes. So it was an eye-opening experience, and one that I'm really fortunate to have guided by Dr. Jennifer Dahl. So she was also a first-generation undergraduate student, went on to get her PhD while she was pregnant as well, and was my undergraduate mentor. I was lucky enough at Eau Claire to be part of a program that invited students to take part in undergraduate research their freshman year, and I mustered the courage to talk to my organic chemistry professor at the time. He said what I expected to hear, which is that no, he wasn't taking any students in his lab, but Jennifer Dahl happened to be right behind him, his wife. And she told me to come after class. They were looking at some silver nanoparticles in the electron microscopy room. So that's a photo of me in that same room looking at the electron microscope. As you can see, it towers above you. Had never seen anything like it before. And I'll be honest, I, at that moment, thought that if I didn't do something with a microscope, all of the sacrifices of those bugs and plants in my neighborhood would be for nothing. So I ended up working with Dr. Dahl for 4 years on gold nanoparticles. But eventually, I came back to biology. My family has been impacted by chronic illness, several family members in particular. And if I was going to go on to do a PhD program, I really wanted to do it in something that felt motivating for me. And human health was really becoming that main motivator. So I thought about how I could use some of my experiences in material science to do just that. So my project was actually on single administration vaccine technology. How many people here have received a vaccine? Hopefully everybody of some sort or the other. So you're familiar with the typical regimen. If you have the COVID vaccine, you keep just getting booster shots every year. But if you're a child and you're getting like a malaria vaccine, or excuse me, a diphtheria vaccine or measles vaccine, it might look something like this. You get your first shot, and then you get another shot in about a month, and then a month after that you get another shot. Well, that's the current standard, but it's really challenging for people that live in places where healthcare infrastructure is not well developed. In those places, the only time a child might be in the hospital and have access to a vaccine is when they're born. And so if you're missing out on these extra booster shots, you're not getting full immunity, and there's a huge issue right now in the world globally where people are under-vaccinated. So we were developing technologies, and I was focused on this bottom one in particular, using biodegradable polymers to deliver the complete vaccine dose in a single shot. So some of the polymers would have a thin shell, some would have a thicker shell, and these were biodegradable. They would degrade over time. One would release at 4 weeks, another would release at 8 weeks, or any tunable profile. So this is really how I started to get interested in this problem of, science can actually have an impact not just on, targeting or solving interesting problems generally, but can also have an impact on making technology more available and affordable for the world. Really quick, just to highlight, this is a video that will play. It's playing. Okay, I can see the numbers there. You can't really see much going on, which is good. Really quick, this is the actual way I was making these particles, which you can see on the right side. They're filled with a test protein, which glows fluorescently under a microscope. You can see the shell there as well. And we're using a technique where we created a really strong electric field and that focused the meniscus into a cone jet, and allowed us to make these really tiny particles that you can see are a couple of microns large. All right, so after grad school, my interest in human health really continued, but I became more aware of some of the challenges of how do you translate technology out of the lab? You come up with this an amazing solution. You know, I was developing and working on these single administration vaccines, but I didn't have the money to take that forward. And I entered the space of venture capital. And I did that in part because I really wanted to learn how these investors were thinking about what makes technology commercializable, and how I can better utilize that information, that business knowledge and insight to better support technologies that I cared about. And I was grateful to learn that even though I definitely expected it to be a bit like Shark Tank, it's not quite that intense. Okay, how many people here have seen Moore's Law before, this concept? Okay, I see a few. For folks that don't know, Moore's Law is just an observation that about every 2 years, the number of transistors on a microchip doubles. And this is a really powerful effect. It's allowed us now to have extremely powerful smartphones. It's allowed us to have AI tools like ChatGPT because we have this incredible computational power, and it's become cheaper and cheaper to do this type of computation. Well, there's something called Eroom's Law that is exactly the opposite. And it's also really just spelled oppositely, spelled backwards, Moore's Law. And this was coined, this term, by Jack Scannell and his colleagues to describe an unfortunate trend that was going on in drug discovery and development. So this blue line here is Eroom's Law. And again, it's plotted against Moore's Law. As you can see that we're getting better and better, and here it's measured as calculations per kilowatt hour. Basically, we're getting better and better at doing computations more efficiently, or packing transistors on chips more efficiently. Well, funnily, in the biology world, or the drug development world, we seem to be getting worse and worse at developing new molecular entities. So the NME up there basically stands for developing new drugs. And as you can see, we're at a point now, we reached in 2000, where we're spending over $1 billion and have been spending over $1 billion on average on new drugs for over 20 years. And that's not sustainable. What are some of the challenges? All right, how many people have listened to this album? Okay, great, fantastic. It is phenomenal if you haven't. But Eroom's Law comes with 4 kind of fundamental pillars that describe some of this phenomena of why drugs are getting more and more expensive. I'll be honest, in the original report, they call it better than the Beatles. I'm going to call it better than Beyonce, though. I think she's at that point where she can be compared, at least in my opinion. So what is the issue? Well, you can think about it like this. If every new song that came out on the radio had to be better than some of Beyonce's best classics, we would not have a lot of new songs, would we? And the issue is cropping up in the drug development world, where the rising standards for drugs, because we're developing efficacious drugs, is moving higher and higher. And so it becomes harder and harder to justify the risk of trying to develop a new, better drug when there's already a drug that might work pretty well in the first place. So that's the first challenge of 4. There's another one. They call it the cautious regulator. Now, this one is not necessarily bad. But the FDA and other regulating agencies, when they see adverse reactions, when drugs are pulled off the market because we learn more about their toxicity and their lack of safety, the regulations around those drugs tends to tighten. That has the unfortunate consequence of making drug development more complex and more expensive. And patient safety now really does come first, and it should come first, but we have to continue to balance that against the unmet need for certain diseases. All right, the third pillar is called the throw money at it problem. And this is describing the idea of pharma companies with their immense resources tending to throw a lot of human capital, in particular, a lot of people at problems to try and solve them faster. Well, I don't know if anyone that's worked in the drug development or drug discovery world knows it's a very, very complex process. And when you throw a lot of people into a very complex process, more often than not, you create inefficiencies. And finally, we have this issue of brute force basic research drug discovery, where pharma companies have invested a lot of effort in trying to screen more and more compounds, developing more automated tools for high throughput screening to get through these compounds, but it's having diminishing returns. We're not seeing the effect of the scaling, and so it begs the question that a new approach is really needed. All right, so these are for, this describes the concept of finding new molecular entities and new drugs. But I do want to spend some time just talking about some of the other issues and thinking about what about drugs that have been around, where their patent, for example, has been disclosed for over 100 years, like insulin. What about these drugs? What's going on here? Well, they're still getting more expensive. 140% increase in insulin prices in a 9-year period for a 30-day supply. Insulin today is still, over the $400 mark for this 30-day period if you don't have insurance. We'll do a little quiz here. How many people think the insulin patent, the original patent, was sold for under $100? Raise your hand if you think it's A. Okay, see a few hands. How many people think it was $100 to $10,000? All right. And how many people think it was over $10,000 for that original patent for a life-saving drug? All right. All the people that said A, you would be correct. The insulin patent was sold for $1. It was sold for $1 specifically because the scientists wanted to make it available to the world. So really, I highlight that. I highlight that because I don't want to overpromise. Science can have can make a tremendous impact on drug discovery, but it's going to take a combined effort in policy, in legal, in commercial regulation, and in the regulatory bodies themselves to create incentives that drive down the cost of medicine, particularly in the United States, especially for life-saving medicine that's been in existence for a long time. But like I said, technology can help, and that's where I focus, so let's get back to it. All right, how many people here have had beer before? Okay, must be a lot of freshmen in the audience. Okay, sure. Why am I calling out beer? Well, beer is really interesting, not just beer, but wine as well. Beer is one of the oldest examples of humans taking advantage of microbes or microorganisms to produce a useful product. You can argue about the utility of beer, but a useful product that they want to use. And it's a 6,000-year-old process, or over 6,000 years old. Well, in the modern era, we're taking advantage of microbes still. And we're not doing it all that differently, although now we can design DNA that can encode for the exact therapeutic protein we want to produce. But it is a complex process. This is the first step. I'll show you a little bit more later, but you have to then do fermentation, you have to purify, you have to package. But again, using fermentation and using these microbes to make recombinant proteins of interest has been life-changing. It's led to drugs like Keytruda. Anyone that has been unfortunate to know a family member that is experiencing cancer will likely know Keytruda. It's an immunotherapy, it's a recombinant monoclonal antibody, and it saved thousands of lives, if not millions. But one of the issues with these types of drugs, they've been wildly successful, but they are expensive. And even when they go off patent, the cost savings, Keytruda will go off patent in the US in 2028, but in China it's already off, and the cost savings are really, really limited. Only 20% here. So Keytruda, again, a very expensive treatment, almost $200,000 per vial, will go down to $153K. So, you still might have to mortgage your house or refinance your house. But if we look at generics, and this is again an incredible medication, Truvada. It's for HIV prevention and treatment. It went generic recently. And the savings you get from a small molecule going generic can be up 98, 99%. We're talking about a drug that was $2,000 becoming $33. And so there's this big question of, can we make traditional biologics and biological manufacturing, so making those recombinant drugs like Keytruda, can we make that look more like small molecule synthesis? And again, this is another kind of shorthand way to describe all of the steps needed in biologics manufacturing. And while this is a simplification as well of chemical synthesis, it's truly an endeavor to create these recombinant drugs and make them safe and stable. And so my first project at Anzu Partners, the investment firm I was working at after graduate school, was working with and evaluating a technology and supporting a technology coming out of, does anyone know what this campus is? It's a lab. If anyone's seen Oppenheimer, this is now what Los Alamos National Laboratory looks like in New Mexico. So there are two scientists there. They do more than nuclear research now, thankfully. There are two scientists there that were investigating extremophilic bacteria. And they were really interested in identifying some of these novel gene clusters that were encoded in these bacteria to see if they contain any new antibiotics. And what they found was that they needed to develop a new system to test whether or not these extremophilic bacteria actually produced antibiotics, because they would only do it under certain circumstances. The other challenging part about trying to produce antibiotics in cells is that they tend to kill the cells, especially when you're using bacteria. And so they developed a process where they took just the cellular machinery necessary from a geobacillus, which is a type of extremophilic bacteria. They used an extract of that cell, so you just had like the ribosomes and all the cellular machinery. And they used that to test these novel gene clusters and found a novel antibiotic. But they noticed one other thing that was really interesting. They weren't the first people to ever try using a cell extract, right, which I'll describe here, to make these novel gene clusters. But they noticed how stable the cellular extract was because they were using thermophilic bacteria. People had used E. coli before. E. coli is a really common organism that's used right now. It makes some drugs that you might have taken. I think E. coli can make insulin, for example. But if you want to use an extract-based approach, E. coli is really, really unstable. And so cell-free protein synthesis offers a lot of potential advantages over what I described before, which is having to work with cells. And maybe just to ground it in an analogy, you can think about cellular fermentation, so using the entire cell to make these proteins and trying to purify it, as a factory that's optimizing for many, many products, and you're trying to force it to only make one. Whereas cell-free protein synthesis is like a custom assembly line that is laser focused on one thing, and that's transcribing DNA into RNA, and then translating that RNA into whatever protein you're interested in. And so it has benefits in safety. You don't have to use viruses now or transfection agents to deliver that DNA into the cells. It's a lot more stable as well. Some of the issues with making Keytruda, for example, in cell lines is that those cells can mutate over time. And those mutations can affect the quality of the end ingredient or the end product, like Keytruda. And so you can have a reduced efficacy in your actual product. But in a cell-free synthesis, you have perfect control. There's no mutation going on. You can accelerate the design, build, test cycle, right? You have this new protein you want to test and sell, or this new therapy you want to test. But traditionally, you'd have to take many, many months to develop a cell line in order to make the protein and test it in animals or test it in whatever model system you want to. Here, you can do it in less than a day. It has capacity benefits, especially in a pandemic scenario. It can take a long, long time to transfer production cell lines to make new products. So if we have to make a new protein vaccine again or something like that, transferring those cell lines to make a new protein vaccine can take many, many months, if not years. If you have a cell-free extract-based system, it could be as simple as changing the input DNA that will transcribe again into the RNA that will translate into the protein of your interest. And it can also be more cost-effective as well, because you're not wasting time and energy on producing other proteins that you're not interested in and having to filter all of them out when you make your product. But there are a lot of challenges as well. And this is where the scientists at Los Alamos recognized there was potential. The traditional challenges were in sustaining the reaction with E. coli. It just wouldn't run for more than 4 hours typically. How do you scale this, right? These are Eppendorf tubes, which are really tiny. It seemed to be really hard to scale it in a batch reaction. And then another really big challenge is how do you make complex proteins like Keytruda, like antibodies? Well, they came up with 3 key innovations. First, again, they isolated bacteria from, isolated thermophilic or extremophilic bacteria. The big advantage there was that the cellular machinery that they had was much more stable to a variety of reaction conditions, unlike the E. coli machinery they were using before. They also tagged all of that machinery, right, with a His6 tag. This allowed that cellular machinery to be easily filtered out. Anything with that tag, the His6 tag, could be collected very easily in a column, and you just get your product of interest in a single purification step. They developed a continuous hollow fiber reactor, which allowed for raw ingredients to flow in while they continuously collected protein, so no more Eppendorf tube, no more batch reaction. And finally, they developed a cheap, long-lasting energy regeneration system. One of the advantages of cellular fermentation, actually, is that glucose, or sugar, that the cells use is actually pretty cheap. ATP, on the other hand, is actually quite expensive if you're trying to get pure chemical ATP. So them generating developing a phosphate donor to make that ATP was another really important innovation. I'll go through this a little bit more quickly. This is just describing the fact that they were able to make all of these proteins and tag them appropriately. We were able to make a test protein, in this case, glow fluorescent or green fluorescent protein, GFP, and it's a great protein of interest. Here you can see this is just measuring fluorescence coming out of the reaction mixture and sustaining over 8 hours in this case. And the coolest part was thinking about taking a traditional fermentation center and being able to miniaturize it into something like this, a 1.7 milliliter hollow fiber reactor. And what we did with that initially is just a proof of concept working with a collaborator was produced 600 doses of a tetanus toxoid vaccine in under 2 hours. And if we wanted to think about making this GMP compliant, right? GMP is good manufacturing practice. It's essential to make sure that these drugs are sterile. You could just put the system in a glove box, which was probably about this big and this high. So again, avoiding saving a tremendous amount of space and allowing for this type of manufacturing system to be drop-in. Oops. And so this progress was going well. We were evaluating the technology, testing proof of concept, showing how cell-free protein synthesis could really revolutionize some of the biologics manufacturing that had run into a lot of challenges. And then March 2020 hit and the COVID pandemic along with it. And so we wanted to pivot. We thought about, well, if we can do this, again, go from DNA to RNA to protein, can we optimize for just producing mRNA to make a vaccine, or to support folks like Moderna and Pfizer that were making mRNA vaccines? And it's a little more complicated than just stopping at mRNA. There are a few modifications you have to make. But in less than 9 months, we were able to develop a new capping enzyme, which helps with stability, transport, and translation of these RNAs to make sure that you're actually getting that they're producing the protein of interest, in this case, the COVID spike protein, for example. And we also improved and made custom enzymes for this blue part here, which is polyadenine tailing. So these enzymes, again, they're just really important to make sure that you can actually translate that you're actually translating the RNA. And we also found that, again, the same approach of a continuous reactor instead of a batch reactor was allowing us, at least in our proof of concept scale, to develop RNA in a third the time and half the cost with greater integrity. And so we saw successful production of a novel multivalent vaccine for COVID in under 12 months. And while we didn't take this forward, it gave us tremendous confidence in working with other partners that were producing these types of vaccines. And today, the company, the scientists, it's now called NTX, or Nature's Toolbox. They're working with government agencies like the Biomedical Advances Research Authority for Development, BARDA. It's a huge funder of research against infectious disease, to deploy their platform and scale it up for RNA production to respond to the next pandemic. And they're working with folks like Ginkgo Bioworks out of Boston, who are leaders in cellular fermentation, to advance their cell-free protein synthesis to make more complex proteins like Keytruda. And these are the two founders here. I caught a photo of them with a giant pair of scissors as they were doing their ribbon cutting ceremony in Rio Rancho for the new facility in New Mexico. All right, so we talked a little bit about some technology addressing some of the challenges in manufacturing these medicines, but I want to shift gears, go back to drug discovery and development, and think about some of the challenges here. So the tools one of the challenges, first and foremost, is that the scale and cost at this stage, even though we've invested in a lot of automation to do this, to look at 1,000 drugs against a target or a disease of interest, You can still only ask really simple questions here, like, is the cell alive or dead? Or did my compound of interest bind to the target that would affect the disease? And when you get a little bit further down, animal studies and a suite of expensive instrumentation are really required to start unpacking the potential toxicity of these compounds, as well as the function of these drug candidates. And the bar is really high, again, for new drugs coming out to be safe and effective. And scaling just this traditional method without better understanding function or having more measurements is a challenge. And so it begs the question, again, can we develop something more effective that allows us to collect more information initially, but is also low cost? So I'll walk through this quickly. Early stage drug discovery, right? As I described, you take like a cell, or you can take a protein. You're going to add all of your different compounds of interest to a microplate and maybe some media. You have to then incubate that for several hours, if not more. And then you'll use some reporter assay. You can use a Firefly, some type of fluorescent reporter assay typically, like Firefly luciferase. And you put in something called a plate reader that basically measures the luminescence that shows you, okay, did your molecule of interest do the thing that you wanted it to do, that you think affects the biology? But there's a lot of challenges here. It's a single parameter. You're only measuring a single parameter here, what that fluorescence assay is doing. You're not getting any information about how the cell dynamically responded to the compounds of interest. Did the cell start moving more slowly? Did it start moving more quickly? Right? All of these things that can be early indicators of toxicity or function. They're endpoint measurements, so again, you have no kinetic profiling, you don't know what the cells are doing over time. And these assays are typically label dependent. So you have to add a dye or a fluorescent marker, which can impact the biology as well. All right, how many people, again, here have a smartphone or a DSLR camera? I would be surprised if everyone didn't just raise their hand. All right, so the technology that's powering your smartphone camera, powering a DSLR sensor, and other aspects of modern day technology like laptops and computers is something called CMOS, or complementary metal oxide semiconductor technology. Basically, it's just a way to make a really tiny circuit on a silicon wafer. And so there were a few scientists at Harvard University that I worked with starting in 2020, 2021, that were asking a really interesting question. Can we use this mature, low-cost technology to better measure cells and collect more information? And so this was the prototype that they built. It's not very pretty, but they packaged a CMOS electrode and a sensor array into some plastic, and they created a little well, which I'm circling here, where they could seed cells and watch them grow over time and see what happens. Okay, I'm gonna walk a little bit through the working principle here so we can kind of understand what's happening. Because, and I think the best way to describe it is that we start off with an array of electrodes. And these electrodes, these little dots here that you can see, they exist under the cell sheet. And they're about the same size of the cell as well. So you can see all of these green outlines are cells themselves. And so you have about at least one electrode per cell in this case. And under each of these electrodes is a set of configurable electronics that allows you to either apply a voltage, it can be an alternating voltage, you can apply a current, or you can measure a voltage or measure a current. So those are the things that we're measuring, and you can almost think about it like a microscope. So how do we see cells in a microscope? Well, we're taking advantage of the way that those cells interact with photons or light. In this case, because we can measure current and voltage, we can start to understand how the cells interact with these electric field lines that are created when you apply a voltage. And the way that those cells interact with those field lines gives you insight about what's going on. And so here we can think about measuring current. This is an image. This is not an optical image. It's just it's what you can reconstruct as you get data from the CMOS electrode array. When there's no cell, it's a pretty even distribution. When there is a cell, you get a big difference. You see that the cell, when we're looking at current, measuring current around where the cell is, the current becomes more concentrated. And to prove that this was measuring actual cells, if you look at a culture of cells, all these red dots are cellular nuclei, And you can see that the current has a maximum where the cellular nuclei are. But it's not very high resolution. So if we want to improve the resolution, I won't spend too much time on this, but you can use impedance instead of current. And you measure impedance in an array. All right, I promise this is the last slide before we get to some of the cool data here as well. So if we apply an alternating voltage at a low frequency and look at those vertical field lines coming off of the electrode that you saw on the previous slide, we can start to understand how tightly the cells are packing. If we apply a really high frequency alternating voltage and look at the electric field lines, those field lines can go a little bit deeper above the cell, further above the cell, and we can start to understand how rounded or flat the cells are. And if we look at the field lines that are almost perpendicular to the electrode array, that are really flat, we can see that the cells are blocking them. And if they're blocking those electric field lines that are lateral, then we know if the cell is attached or detached or how high it is off from the electrode. All right, so what that allows you to do is start building some really interesting images, or electronic images. These are not optical again. Where you can start with no cells and you just have a blank sheet. You start adding cells after 2 hours. You can see that there's a bit of cellular flatness, right? So the cells are not, there's no intercellular junction strength, but there's a lot of cells that are just chilling on the sheet. If we look, and there's still a lot of detached space in blue. We start to see now more yellow popping up, which shows that there are cell-to-cell interactions, and the cells are still pretty flat. As the cells continue to grow here, you start to see that the cell-to-cell interactions, this barrier function in red, becomes the dominant force. So we wanted to think about, all right, can we miniaturize this, run this in parallel, make it applicable to high-throughput screening? The answer was, thankfully, yes. And again, this was just taking advantage of the same technology that we use to make smartphones and camera sensors. Now, one of the cool things is that this, if I go back really quickly, this microplate, you can measure all of this in the incubator. You don't have to take it out. It will continuously monitor what those cells are doing as long as you're looking at the impedance or the current or the voltage. So it allows you to have these long-scale dynamic measurements. And if we do a simple toxicology assay with a sample cell line, you can again see these are not optical images, but you can very, very clearly see that before the drug is added, you see the cell sheet operating, you see the cell sheet completely covering the electrode matrix in all of the wells. And after you add the drug, you can see based on the concentration, the highest concentration is over here on the right. It kills all of the cells. In a lower concentration, you're seeing more spotted blueness, which means that a lot of the cells have died, but not all of them. And what we can start to do is build some really powerful dynamic videos. So I tried to code this to make it a little bit easier to understand, but we have a variety of cell types here. We have some cells from kidneys, from the brain, from the colon, from the lung, from breast tissue. And what we can do is compare how these cells grow. We can look at how tightly they attach to each other, if they're very red, like cells in the colon that are epithelial cells that need to form tight junctions. We can again look at cells in the kidney that also need to form tight junctions. And we can see cells that are less interested in forming those tight junctions because that's not fundamentally their function. So one of the big things you can see here is that without any labels, without optical measurement, we can start unpacking bits and pieces of cellular function. We can see the cells grow over a time scale. We're getting a tremendous amount of information compared to an endpoint optical or an endpoint fluorescence assay. We can start looking at how a single cell line responds to compounds. So these are cells that are just growing evenly. There's no compounds here. You can see they're all happy. We add the compound. We start seeing some changes. We see one here immediately where cells are detaching. We start seeing detaching over here as well. In this video, in this bottom left-hand corner, if you look again, the compound we added here to A549 cells, which are adenocarcinoma cells, they're a type of cancer cell isolated from the lung, we see if we add paclitaxel, which is a chemotherapy agent, it's having an immediate effect on these drugs, or on these cells, especially in a high concentration. We can start to look at other cells. These are MDCK cells, they're kidney cells, so they form really, really tight junctions. And we can see some different effects. We know what some of these drugs do over here. This one is highly toxic. But some of these other drugs, like the one up in the corner, cytochalasin, is known to disrupt microtubule formulation. And so instead of just killing cells, for cells that form a tight, I'll get back there one more time, a tight junction, like MDCK cells, like these kidney cells, we can immediately see the effect of adding something that would disrupt those cells' ability to actually form those tight junctions by affecting the cytoskeleton. And so there are immediate implications here in inflammatory bowel disease, where this really rich barrier function data set can help us understand functionally what's going on when we're looking at drugs in a high throughput screening sense. And this has been a huge area of focus for early pharma partnerships, because this has traditionally been a very tough disease to understand, looking at ulcerative colitis and Crohn's disease in particular, because we don't have good functional tools to measure them at scale. And we can also start building these compound screenings, where we're looking at all of these different parameters in a single run, without any labels again, in a single experiment. And we can start building maps that identify functionally where compounds land. And so when we know the known bioactivity, for example, this light green here is cytoskeletal signaling. We have different ways that different ways that these drugs, all these blue dots are drugs interacting with these cells, cause toxicity. If we create a map of known compounds and their known effects on drugs, when we're looking for new drugs, we can immediately start to classify them as toxic because they're causing DNA damage, as CDK inhibitors, for example, causing DNA damage, or as disruptors to cytoskeleton formation. So this $250 smart plate can feasibly start to replace over $750,000 of instrumentation. And the big promise here, and the thing that I'm excited about, is thinking about how this could be used to accelerate drug discovery, saving companies up to 5.5 years and $400 million per drug. So we can think about what the future looks like here. I showed you a lot of images before. So this is their next-gen microplate. You can see the resolution is much higher. You can start to actually see individual cells here instead of what looks a little bit more pixelated. So the company is working on scaling these types of wells up to provide even richer data sets without increasing the cost of the instrument. We're thinking about measuring cellular metabolism as another aspect of this process, looking at measuring electrogenic cells like neurons and cardiac cells because we're measuring current and voltage, and those are directly applicable to these types of cells, and we're trying to scale up as well. So I'll highlight the team here. These are the three founders, Jeff, Dwayne, and Vince, myself and another investor. They were at the Society for Lab Automation Screening. They won the New Product Award this year as well. And so I'll just end really quickly by saying, with new technology, scientific approaches, and collaboration, I do think we can close this gap. The work that Cytotronics is doing to really fundamentally develop new ways to functionally understand what drugs are doing at a high scale. What NTX is doing to improve the cost, the reliability of drug manufacturing are two key drivers. But again, it's going to take this combined effort to ultimately close this gap, and it's going to take a lot of what you're doing early stage fundamental research as well, developing new methods and tools to identify drugs. So I will pause there. Thank you so much for having me. I really appreciate it. I know I'm an esteemed company of a list of speakers, and I hope you took something away, but I really want to make sure there's time. Hopefully there's time to answer a few questions if anyone has any thoughts, anything that didn't make sense, anything that looked interesting. Let me know. I think there are 2 mics up here, so feel free to come up. No pressure, of course. All right, see one.
You have to forgive me because I'm an organic chemist.
No worries.
The cell-free protein synthesis that you were talking about.
Mm-hmm.
So that's some jargon that I'm not particularly, adept at. But what do you mean specifically by the cellular machinery that you're talking about? So what is that specifically? But also more to the point, does that cellular machinery that you choose, does that change depending on the protein that you're synthesizing? And then how do you decide what cellular machinery you use from the target that you're looking at?
Yeah, a great question, and thank you. So the cellular machinery we're interested in is just the proteins and enzymes that are responsible for taking DNA, transcribing it to RNA, and the proteins that are responsible for translating, and the ribosomes, that are responsible for translating that RNA into the protein of interest. So instead of having a cellular nucleus, instead of all the other organoids that you have in a cell, we're just focusing on those fundamental pieces. And there have been folks exploring a different variety of cell-free protein synthesis. Some have tried using eukaryotes. Because of how complex eukaryotes, it's really hard to isolate those components. The advantage, though, is that they can produce antibodies. Eukaryotes are naturally producing antibodies right now in our own cells. So what we're trying to do is use a cheap platform, like bacteria, really easy to make those proteins, and it's a simpler cellular machinery set, but using the right bacteria. So instead of E. coli, using thermostable bacteria, or thermophilic bacteria, that are less likely to peter out in the reaction because the temperature is not quite right. It's really, really robust machinery. And then the final part of it is figuring out, and this is what a lot of the work they're doing with Ginkgo Bioworks right now and some other collaborators, is thinking about, can you take that cellular machinery from bacteria, add some specific modifications that can allow them to start folding more complex proteins like antibodies, but still maintaining that cost advantage. Because otherwise it's not, there's no point in doing that work. If you can't maintain the cost advantage, you're better off just using cellular fermentation. So that part is a really hard challenge, but there's a lot of work right now going on with their enzyme engineering to see if they can better fold more complex human-like proteins. Thank you.
Thanks.
Hey, what's going on? Okay, so those microplates, like, do they give like a live like feed of how like cells and things like that are like affected? But like, can you like go into more detail about that?
Yeah, they are providing they're measuring in those videos. It was sped up, so they're measuring the cells every 5 minutes. But the cells don't move very quickly. They don't divide very quickly. So you can build up a time-lapse series to make videos like that. So yeah, one of the big advantages, again, is that when you're typically, when you're using a cell microplate or you're running an experiment, you are adding your drug. You're then putting it into the incubator to see how that drug interacts with the cell, right? But you don't know what's happening. And then you have to take that microplate out, look at a microscope, or take it to a plate reader to find out what happened. In the SmartWell plate case, because you're constantly measuring what's going on with the cells while they're in the incubator, you can already start to see how those drugs are impacting the cells. So you are getting functionally a live feed that you can access in the cloud. They're using cloud storage for that. It's a lot of data. But that's the type of information that folks and scientists and collaborators are working with are really excited about, this live feed and dynamic information.
Okay. What's the difference between that and like a microscope?
Yeah, so a microscope, the challenge there is it's really hard to scale. How do you look at all of the wells at the same time? And the other aspect of it is the cells need to be grown in a incubator. That incubator is held at 37 degrees. It has controlled oxygen and carbon dioxide. It's really allowing the cells to grow at their most optimal conditions. So it's really hard to fit a microscope inside an incubator as well to try and do those measurements in real time and to try and move the cell plate under the microscope constantly. It's a really difficult automation challenge that folks haven't yet solved because it's too expensive. So in this case, because you don't have to add any additional instrumentation to get that live image, it's a more viable way to actually see what's happening at the same time. But it's very similar to a microscope, just miniaturized in a major way.
Okay, all right, thank you.
Yep, thank you. Appreciate the question. All right. Any others? Raise your hand. Okay, perfect. Thank you so much. I really appreciate the time. Feel free to contact me. I'm going to be here for a little bit after. Oh, yeah, please.
I do have another question.
Yeah, sure.
This one's not scientific.
Yeah, it doesn't have to be scientific, by the way. It can be anything about my background.
No, so I do have a question, and I want to do a thought experiment. So let's say that you're talking about technology is what this was about. And let's just do a thought experiment and say that you're able to take a drug and reduce the cost from whatever it is, say 90% reduction. In your opinion, how much of that savings, based on all the complexity that you're talking about, goes into the cost of drugs, how much of that 90% reduction would the consumer see? Or even if you can go 99%, we're not going to see the full amount. Because there's a lot of other stuff going on. But what's your thoughts about that?
Yeah, I mean, I think where you can see those big reductions is in generics in particular. So, for example, let's say the NTX process, like you said, that cell-free process allowed antibodies to be produced at 1/10 the cost. Any new antibody produced using that system is going to be patented. For 20 years, it's going to still be super expensive. But as soon as that patent expires and other manufacturers can start producing that protein or that drug without infringing on the patent, that's where you can actually start to see the savings. And that's why I called out that Truvada example, where you can go and buy Truvada for $33, or you can buy it for $2,000 if you use the brand name developed by Gilead. And it was $2,000 for 20 years, at least, with that brand name. So I think the real promise is not going to be in new drugs that are coming out immediately, but in a lot of these really prominent immunotherapies, again, like using Keytruda, Humira is another one. As soon as they go generic, if we have technology that can actually that can be more cost-effective than just the cellular fermentation, which just has a limit on how much you can reduce the complexity there, I think that's what I'm excited about in terms of really reducing the cost. So you can see, Keytruda go from $100,000 or $200,000 to $20,000. But yeah, I think it's going to be the generics that make a difference first, not any of the new drugs for consumers.
Okay, thank you.
Appreciate it.
All right, Tayo, I have a question for you.
Yes.
Well, your work is very interdisciplinary.
Mm-hmm.
Will you speak just a bit about how that journey kind of sparked all of this work together?
Yeah, I think it's two parts of thinking about interdisciplinary work. Part of it is I've always been interested at those intersections because, again, broadly, science has always interested me. But from an investor perspective or from a scientific development perspective, those intersections, those interdisciplinary programs or approaches like cytotronics, where you're taking semiconductor technology that has been used for smartphones, displays, and cameras, and applying it to biology, more often than not, that's where some really interesting scientific innovation and insight comes from. And so I encourage all of you to really challenge your assumptions of what you can do in your field. Think about using technology like CMOS integrated circuits potentially, what you can do with electronic circuits. Find an electrical engineer to do the hard math for you. But you can use the biological concepts to say, hey, this might be really interesting for measuring this type of cell because I know the cell will interact with, again, the electric field on a plate, something like that. But again, I really encourage the interdisciplinary approach as an investor, as I've seen a variety of technologies, some of the most successful ones, some of the most interesting ones, and some of the most impactful ones are technologies where biologists, chemists, and engineers are coming together to build something that you wouldn't immediately think of as a viable solution. Yeah, one more question.
It's a longer way down than you think. The figure that you showed with Moore's Law and Eroom's Law, it's a great figure. So that gap, right, between, what was it, cost of discovery? Yeah, there we go. That gap.
Yeah.
For the researchers in the audience and the aspiring researchers in the audience who find this really interesting, because we have a lot of students here who are excited about human health and possible ways to advance. If you were going to try to identify, so it's a lot of space in that gap, and there's probably just a ton of things contributing to that gap. If you were going to try to identify two key areas that make up a lot of that gap, what would they be?
Well, maybe I can think about two key areas that make up a lot of the gap, and maybe some other promising technologies or approaches that could help close it. Again, I think it's a lot of it is those pillars that I talked about before, where it's becoming harder and harder to make better drugs. And the target-based drug approach is not the only one we can take. If we think about phenotypic screening, where you start with a disease pathway, but you don't know the target, or you don't know all the targets, there's incomplete knowledge, and screen compounds against that, and then later identify the target as an approach. I think that type of work is going to be really impactful in terms of broadening the ways we can discover biology. We don't have to know the exact target up front. But I think if you actually look at this graph, one of the other opportunities that comes out is how do we take advantage of the fact that computation is getting really, really cheap? How do we use this? And I talked about it earlier in class today, but I think again, of course, like certain AI models can be really useful. Large language models, in particular, have already been able to produce a brand new protein. There's a great paper if you look up ESM3. It's a large language model that was designed to take queries about protein design. And in this paper, they describe developing a brand new green fluorescent protein that doesn't exist currently in biology. That's only 58% similar to the next closest green fluorescent protein, and would normally take 500 million years for biology to develop that type of protein that was that dissimilar. So I think there's tremendous potential when thinking from a technology perspective and taking advantage of all this computational power. Again, some of the challenges are the fact that a lot of AI models are black box. We don't understand how they're making their predictions. There's too much siloed data. Each company has their data set, or a hospital has their data set, and they want to monetize that or create value for themselves, so they don't share. So there's a lot that we need to do in creating standard, large, diverse data sets for AI to take advantage, but that's what I'm really excited about. For the folks here that are not technologists or engineers or chemists, and more interested in public health in general, there's a tremendous amount of work you can do in the political space, right? The FDA is a huge driver of what drugs we develop and how easily we develop them. And again, in a regulatory space, also thinking about what limits should we be exerting on pharma companies where they charge Americans $200,000 for a drug, but they charge Europeans $100,000. So there's a huge room to play, no matter what your discipline is here, in addressing this gap, which is also exciting, but also slightly daunting, but speaks to the kind of interdisciplinary approach, the amount of collaboration that's needed as well.
Thank you, Dr. Sanders.
Yeah,
we'll let you
you.
go. We appreciate your work here and all that you're doing for
thank
our
you.
society. Thanks.
Thank you.